Good evening, everyone. So, welcome to the Van Bug. And as usual, we have two fantastic speakers today. Then we'll have some networking session and with free pizzas and beers. So first of all, let me introduce Maia Smith. She did her undergrad from SFU, and then she was a bioinformatics master student in Dr. Sorosov's lab, and she was working more on data visualization, and she's going to be talking today about her work that she did in Dr. Sorosov's lab, entitled Visualization Methods for Spatial and Temporal Clonal Evolution of Cancer. Let us welcome Maia.
All right, so thanks for the introduction. I'll be talking about the work of my master's thesis, which focused on visualization methods for spatial and temporal clonal evolution analysis in cancer. That's a mouthful, so I'll give a little bit of background first. So cancer is an evolutionary process, as many of us know. Here's a schematic representation of a tumor, a very simplified version of a tumor, and this tumor happens to have three different cell populations. These cell populations are also called clones and are related to one another in an evolutionary hierarchy. So this hierarchy can be described by sort of a tree, which I've shown here. It's called the Clonal Phylogeny, and what this is telling us is that the blue clone is ancestral to the descendants' orange and green lineages. So this is important because this evolutionary process acts over time, and this can influence factors such as treatment resistance. So for instance, we might have a drug application here at a given time point, and this drug might be effective in eliminating the blue and orange clones, but perhaps the green clone has a mutation that enables it to continue proliferation, which is what you see here, and perhaps it even continues mutating and eventually gives rise to a descendant population of cells, here shown as the purple clone. So cancer is an evolutionary process also acting over the spatial dimension. So as we know, metastases can occur. Let's say that the original tumor was occurring in the kidney, and perhaps one of the clones, in this case the green clone, metastasized to the brain and continued its evolutionary course there. So far we can see that clonal evolution impacts clinical endpoints such as treatment resistance and metastasis, and it's pretty new that we can actually quantify this clonal evolution. However, we may have the data, but we need a way to visualize and interact with this very complex and high-dimensional data type. So that's what I focused my master's on, was developing visualization tools for the visualization of this clonal evolution data. And the first tool I developed was Timescape, which is for visualizing temporal clonal evolution data. So here I'll illustrate Timescape using a patient, an acute myeloid leukemia patient from Ding et al. And this patient had a diagnosis sample, which I've represented here, and a relapse sample. Now showing the same data with the Timescape looks like this. So on the x-axis we have time, diagnosis here, chemotherapy, and followed by relapse. And on the y-axis we have clonal prevalence. Also note on the right-hand side we have the clonal phylogeny again, relating clones to one another in an evolutionary hierarchy. So what we're really seeing in this plot is the red ancestral clone expanding and giving rise to its descendant green and blue lineages here at diagnosis. Chemotherapy hopefully shrinks the tumor, and therefore the plot shrinks as well. And a single new clone emerges from the blue lineage with chemotherapeutic resistance and expands at relapse here. So I'll now illustrate the interactive capabilities of Timescape with this patient. So here we can see a diagnosis. We have a diversity of clones as compared with relapse, where we have a single clone. And you can see that this clone is clone 5, and it's present at 100% prevalence, as noted there. And sometimes it's easier to see the various clonal dynamics of each clone by separating each clone such that it changes prevalence on its own track, as you saw at the end there. Unfortunately, that's gone. So thus far, we have considered within tumor evolution. What about between tumor evolution? When there's multiple tumors, multiple metastases, etc. So that brings me to the second tool I developed. This is Mapscape for visualization of spatial clonal evolution data. And I'll illustrate Mapscape using a metastatic ovarian cancer patient from McPherson and Roth et al. Now, Mapscape was actually used in this paper to visualize all 7 patients, all 7 metastatic ovarian cancer patients. You can go to the paper if you want to see all 7. Here I'll just be showing one of them. So this is the framework for Mapscape. On the right-hand side, you can see the clonal phylogeny again, followed by an anatomical diagram or image. This could equally be a CT scan or any sort of medical image. It depends on the user input, whatever the user would like to see. And so each sample is represented as follows. The anatomical location of the sample is illustrated on the image there. And the sample, of course, has a label. In this case, it's left ovary sample 4. And a cellular aggregate view, which shows the proportionate prevalences of each clone in that sample. So here you can see the dominance of the yellow clone. And finally, the clonal phylogeny, which is the same as the clonal phylogeny in the legend, but only shows the clones that are actually present in that particular sample. So this seems quite simple, and once we add all the rest of the samples for time point 1 in this patient, we can see it gets a little more complicated, but still fairly easy to read. And these samples are all coming from the interperitoneal space. Now, at time point 2, there's a brain metastasis, and time point 3 has the relapse in the interperitoneal space. So altogether, this rather comprehensive visualization enables the analyst to hypothesize about the evolutionary trajectory of this cancer in terms of treatment resistance and metastasis. For instance, this video unfortunately isn't working here, so I'm going to just bring it up here. So for instance, here we can see at the right uterus, there's a green clone, and this green clone is actually responsible for metastasizing to the brain and seeding that metastasis there at time point 2. Now also note that after time point 1, there were surgical interventions in the interperitoneal space, but a few clones survived, pink and yellow clones. They escaped surgery and actually metastasized to the right pelvic mass and the bowel in the interperitoneal space there. So in conclusion, as mentioned, clonal evolution is important to study because it impacts clinical endpoints, and we can now quantify this evolution. And additionally, due to this work, we have a way to actually visualize and interact with this complex and high-dimensional data. So all of the code is open source. Here are the links if anyone's interested. And also I've put the third tool I developed here. I didn't have time to go over it, but here's a screen cap. It's for single cell analysis. And I'd like to acknowledge my supervisors, Saurabh Shah and Sidney Nielsen, many others from the Shaw Lab, and of course NSERC and CIHR for funding. So thanks very much, and maybe we have time for a question or two.
Questions?
Yes.
That's fantastic. All technical questions to you later. But I was curious about these between individual comparisons. I mean, are there sort of predictable pathways of mutation that these are comparing across individuals that they respond to treatments?
Right. So one pattern that was noticed in a paper also coming out of the Shaw Lab is in transformed follicular lymphoma. And transformed follicular lymphoma compared with progressed follicular lymphoma, and there are noticeable clonal dynamics that occur consistently. So for instance, I believe in the transformed lymphomas, where the lymphoma actually presents itself differently at one time point versus the next, it's actually a minor clone from the first time point that expanded to the second time point. So patterns like that we can uncover. In terms of individual mutations that are consistently occurring in certain situations, that's also something that can be studied using this tool. I'm not sure if the studies are out there yet, because I think these clonal studies are pretty new. But yeah, I'm sure that there are patterns to be discovered there. Yes?
So does it just take PyClone's output as its input?
Not PyClone, because PyClone outputs mutation clusters. So you need to take turn the mutation clusters into clones and clonal prevalences. And to do that, you can use sit-up, clonal inference, something.
Is it flexible, or is it only like that pipeline that works with your?
No, so the tool takes in, I mean, it's all documented, but essentially the clone ID and the clone prevalence and either the sample location or time point. So however you get there, it's up to you.
Yeah, so the next speaker over there.